Well, hello and good afternoon, ladies and gentlemen. Thank you for attending our webinar from Rodale Institute on hog management in the pastured system. Today's webinar is going to be focused on pastured pork production, but more importantly, with the research updates that we have gathered over the last year from a recent project. So we're giving you all of those details shortly. But first, I'd like to discuss a few housekeeping items. This webinar runs from 2 to 3, and all registered attendees will be sent a copy of the webinar slides, so you don't have to worry about rushing to take notes or writing down some of the email contact information. The way the webinar is going to be organized, you can see there's the agendas that have it organized into distinct sections that after certain sections, I'm going to allow for questions and answers. And the questions, they could be asked through the chat function of the webinar. I think you'd find that somewhere in your tools. So you could type your questions there. I'll read them off to the group and answer them as best I can. We do have a panelist here, so some of the other project members on our grant are joining us today, and it looks like we have one of them, Dr. Alex Hernandez, here. That I'm going to unmute him for now. Oh, and Dr. Hernandez.
Yes, hello.
Hi. Why don't you go ahead and introduce yourself?
Hi, my name is Alex Hernandez. I am a associate professor here at Kutztown University in the biology department, and my expertise is in parasitology. I study worms in particular, roundworms, nematodes, tapeworms, flukes, spiny-headed worms, acanthocephalans. All things wormy is my interests.
Very good. Thanks, Alex. And I see that Yushi Lee, Dr. Yushi Lee from University of Minnesota, she just joined. Yushi, are you with us?
Yes. Can you hear me?
Yes, I could hear you very well. Thanks for joining. And would you mind introducing yourself to the audience?
Not at all. Yes. I'm usually associate professor at the University of Minnesota Vice Central Research Outreach Center located in Morris. I've been here since 2005 for 14 years already, working on swine behavior and welfare, alternative swine, and organic swine protection.
Thank you very much, Yushi. And I'll introduce myself. My name is Rick Carr. I am farm director at Rodale Institute. I've been with Rodale for about 6 years and joined the pastured pork production and research team about a year, maybe 2 years ago. So I'm really looking forward to sharing some of the research that we've been collecting over the last year and a half. All right, so with that, let's get into it. All right, first I want to say, I want to reference, and I'm going to do this often, our previous webinar on pastured hog production. And you can find that at these websites. It's on YouTube, that you can see the links there, as well as it's in Rodale Institute's website. So at that webinar, there was a great deal of detail put into production management, breeds, pasture management, and systems that we won't cover this time. So I'd like to, I'll give a brief overview of that and just cover some of the highlights. But with the time that we have, I'm just going to reference that and you'll be able to access that. That's, again, available at YouTube and Rodale Institute. All right, so let's get in. Why pastured pigs? I'll say that it's not a novel idea. Probably 150 years ago, that's the only way we did it. And it was like that for thousands of years, probably about 10 to 12 thousands of years that pigs were out on pastures or they were left to roam on their own. But, you know, over the last 150 years, things become more mechanized and pigs found themselves getting less and less time outdoors. So as farmers and Researchers, we kind of have to relearn how to put pigs on pastures effectively. But I'll say at the heart, a pig is still an omnivorous animal, well adapted to foraging. It's going to be a little bit different than other foragers that we put on pastures. But a pasture itself can provide a diversity of nutrients and supplement grain costs. And probably one of the biggest expenses in organic pork production is going to be your grain. So if you could put your pigs on pastures, you're going to receive lots of benefits, and one of them is in lowering production costs. And over the last half a century, there's been numerous research put into animal welfare. When you put pigs on outdoors, either on a pasture or not, but they're not confined, the animal welfare increases. And so you can think of it the opposite way. The more you restrict an animal into a box or a cage, a pen, or concentrate them, you're going to start reducing innate behaviors, and then you develop chronic stress, and that's going to be found in animal life expectancy, and they can develop all kinds of other issues. All right, omnivorous, monogastric. All right, so here's just a general picture of a pig's stomach or the intestines and the digestive tract. So they're similar to us, humans. That monogastric is single compartment. So other examples are going to be chickens and human beings. So they require diversity of inputs, and they're different from a ruminant that is going to be processing the material, processing the pasture differently. All right. Omnivorous, it's going to feed on both animal and vegetable substances. So, in reading some of the historical books, pigs are survivors. They'll eat whatever they can to survive, and that's what has been making them very attractive to human beings. They essentially have co-evolved with human beings for about 10,000 years. Wherever humans went, pigs went, and they provided the meat that required for, let's say, conquering armies into new nations. Where a pig or a cow might take, 18 months to finish and get the meat from. Pig, it's going to be 6 months, all right? 6 to 8 months. All right, breeds and management of pastured pork. Certain breeds, there's lots of different breeds out there. Certain breeds are more adapted to foraging. And I'm probably going to reference this often, but in the previous webinar that Shelby Dukes, a former Rodale Institute employee, she was managing the animal operation here. She delivered that webinar, and she speaks in great detail about certain breeds, but there are different breeds, and a farmer or an owner would have to decide which one is most appropriate for their operation. There are numerous housing, feeding, and watering systems for farmers, and some are commercial. You can build your own. Essentially, you want to provide some structure for the animal for safety and weather, and just to keep them healthier and happier. Yeah, but there are numerous available. Here are just a few examples of watering systems. You could create your own kind of system or purchase big watering bins. And then the feeder bins, they could be just as unique, or throw some feed on the ground if you had to, whatever it takes. But there are many out there, and I think the point is that the practitioner is going to have to choose what's most appropriate for their operation. Pasture management, that's where things begin to vary when it comes to other grazers. The pastures are different. They require a little bit more intensity and a little bit more focus. So 2 examples here. I think the one on the picture on the left, it's clover, and that's probably early spring or maybe late fall. I can't tell from here, probably late fall. And yeah, that's late fall, and that's what we would have. And you would probably have clover pasture or paddock going all year round. And then here on the right is 2 different pasture types. You can see the distinction. One is a mustard, and then can't really tell what the other one is, but they're very distinct, and that's, I think, late spring and that time. So you're managing it for different times of the years, and you might have to do replanting and plowing and taking care of your fields in that way, so that you're providing, you know, the best benefit to your pigs in as far as nutrition. All right, pigs notoriously get a bad rap. You know, we all know the saying, a pigsty, where actually a pig pen is probably the most cleanest of all farm animals. But they do get a bad rap, and I'm going to discuss some of those here. When pigs escape, yeah, it can become a serious problem. Here in the United States, there's states like South Carolina, North Carolina, Texas, and Florida come to mind. They have feral pig populations. And despite all their control measures and allowing hunting 365 days a year, 24 hours without restrictions, they still can't control the pig population. So you can understand why a state, we're here in Pennsylvania, state officials, they resist putting pigs on pastures because of they could escape and then become feral. And when they become feral, you get environmental damage. This is just one of our paddocks here at Rodale Institute, and over one weekend, we were doing a pasture management study, a nutrient content study, and we wanted to know how much a pig consumed. So one weekend, it was a rainy weekend, and over just a 2-day period, they managed to tear up all of the pasture. And after that, the pasture won't regrow. You're going to have to do something else to it. You're going to have to flatten it out, till it out, cultivate it, whatever it takes to get something else growing. Otherwise, you know, weeds or something that you don't particularly want is going to come up. And uniquely, you see there's 4 step-in posts there. That was our exclusion there with a welded wire, not welded wire, but a poly wire keeping the pigs out. So you can see what it used to look like, and it did a good job of keeping them out. But you know, pigs going to forage on above ground and below ground plant parts and whatever's in the soil. And here's an aerial shot of the same time. And on the far left, you could see that beat up area. And even right in the middle of the brown area, that's that little exclosure that I mentioned where they weren't allowed to feed. So, you know, the good thing is exclosures work, but the bad thing is like we couldn't really collect any more data from this, because how do you estimate how much a pig ate when we couldn't harvest the biomass? That's another subject though. Swine pests and parasites. There are numerous ones. Some are easier to control than others, but they seem to be at least one of the greatest obstacles for pig farmers, especially organic pig farmers, just because when it comes to at least parasites, there are no truly effective parasite control measures. And this is coming from, you know, roundtable of extension agents and farmers and researchers, and there's numerous anecdotal data, and one farmer is going to say, we prefer this product, we seem to think it works, and another farmer saying this product, you know, doesn't work at all. So a lot of anecdotal information that what our goal here, the panelists we have here, we're going to focus pretty much the remainder of this on that issue of parasites and trying to address what that means for organic pig farmers in the United States. And so with that, I will open it up to some questions that anybody might have right now. And if those who are coming in late, I did say that I'm going to ask questions or take questions through the chat feature on the webinar. So you're free to give it about 30 seconds, see if anybody has one. And I'll read it off to anybody as they come in. All right, the only chat came from early on was from Stanley from Tanzania. Thanks for joining us from Tanzania, and I hope you enjoyed the webinar. All right, so with that, I'm going to turn it over to Alex Hernandez, and he's going to discuss, he's our parasitologist, and going to discuss a little bit about parasite species and life cycles. Go ahead, Alex.
Thanks, Rick. So what I wanted to talk to you about today was the three most common roundworms that we know infect pigs the world over. And the first one that we have been focusing on in our study is Oesophagostomum, which is a mouthful to say repeatedly, but it's more commonly referred to as the nodule worms. The adult worms live in the small intestine. There are separate males and females. There are strong differences between males and females. In all of the roundworms, females tend to be much, much larger than the males frequently. And these worms mate in the small intestine. The female produces eggs, and she begins to produce eggs somewhere between 20 to 40 days after the host or the pig was initially infected. She'll pass those eggs out in the feces, and outside, those eggs will hatch into an infective larva that can survive in the environment for up to 1 year. Those eggs will hatch into those infective larvae depending on the right temperature and moisture conditions. Once the infective larvae are picked up in the pasture by the pigs, those larvae are going to penetrate the intestinal wall and mature into a larger version of themselves until they eventually reach their adult size and come out of the intestinal wall and live in the center of the gut, the lumen of the gut, where they begin to mate and pass out eggs. There is some limited immunity to these worms, meaning that the pigs will mount an immune response to these worms, but it's not a very strong immune response. And as a result, reinfection can occur for the same host individuals over time. Next slide. The second species of worm that we are studying is hello? Oh, sorry, I thought I lost you there. The large intestinal roundworm, Ascaris suum. These worms are quite large. The adults can be anywhere between 10 to 16 inches. And here too, we see strong differences between males and females. Females are much larger than the males. Adult worms can live in the intestine anywhere between 6 to 9 months. And once they do get into a pig, it takes about 60 to 80 days for those worms to begin to produce eggs of their own. The eggs is what we can actually see in the feces, and what comes out in the feces is an unembryonated egg, an egg that needs to complete development. And that takes anywhere between 3 to 4 weeks, and it's strongly dependent on temperature. The records are that any temperatures above 15 degrees Celsius or 59 degrees Fahrenheit are usually okay for those eggs to develop. The warmer and more moist the environment, the quicker that those eggs can embryonate. And it's those eggs that have to be ingested by the pig, eggs that have the little worm embryo inside of it. And those eggs can actually survive in the environment anywhere between 5 to 11 years. They have a very thick shell that is quite resistant to drying up, and many different kinds of chemical compounds have been tried that can make them resistant to you trying to attack them. Once a pig does eat an egg, that egg is going to hatch and a little larva is going to come out that's going to be taken to the liver first, and there it's going to migrate through the liver, usually creating what are called liver milk spots on the liver. That's because of the little larvae that are migrating through that organ. After they go through the liver, they reach the circulatory system that takes them to the lungs. And in the lungs, they're going to mature into a larger version of themselves after a few weeks. And then they're going to emerge out of the lungs and move down the trachea of the pig back to the small intestine where they began, and then become adults where they'll begin to produce eggs once again. Next slide. The last species that we've been focusing on is Trichuris suis, which is a swine whipworm. And these are roundworms as well that, as you can see in the photograph, are called whipworms because the tail end of the worm is much, much thicker than the anterior, the head region, which is the thin bit that you see there. Those adults are anywhere between 2 to 3 inches long, and they live in the large intestine of the pigs. And inside that large intestine, they can live anywhere between 4 to 5 months. They produce eggs that are the infective stage, and they will begin to produce those eggs anywhere between 40 to 60 days after the pig became initially infected with those eggs. The eggs are very unique in shape. They are described as being lemon shaped, and they have these 2 little plugs at either end. No other egg that of a roundworm or any kind of parasite that lives in the intestine looks like it, so they're very easy to identify. And just like in Ascaris, those eggs come out into the environment in the feces, and outside in the free living environment is where they complete maturation or embryonation. And it takes anywhere between 10 to 12 weeks. Again, depending on temperature, development will occur above 59 degrees Fahrenheit, that's about 15 degrees Celsius. And it's dependent on moisture. The more moist, the warmer the environment, the quicker those eggs are going to develop into a little larva. And then the larva inside the egg waits until the whole egg gets consumed or eaten by the pig. And they can survive in the environment. Those eggs can survive in the environment anywhere between 3 to 4 years. If you can click the next. These parasites actually are one of the 3 species that actually mounts a very effective, well, the host mounts a very effective immune response. And so the worm has a very limited amount of time to find them a partner, a mate, produce eggs, and start expel those eggs out in the feces. Because after about 14 to 30 days, the pig's immune system begins to catch up and begins to attack the worms and expels them. And so reinfection with these worms tends to be much rarer because there's age-related acquired immunity with these infections. So that's an introduction to the life cycles of the worms that we've been studying, and I'd be happy to try to answer any questions that you might have.
Thank you, Alex. Yeah, anybody's welcome to ask their questions in the chat function. I do see that somebody did ask after we moved on, will you go over the organic methods you use for treating parasites? And no, that's not going to be something that we're going to address today. And I could say from Rodale, it's just put into the feed, and it's a diatomaceous earth, and there might be a few other things. Diatomaceous earth tends to be the most common. Yushi might know better than I, but farmers will probably use a cocktail of things: garlic, anything high in tannins like acorns. But no, I mean, you're almost just as easy as just Googling organic methods for treating parasites, because that's kind of all the information that even from science that we know. Okay, well, okay, how long is the life cycle of the roundworm? Go ahead, Alex.
Sorry, I had the mute button there. Yeah, so it depends on the species. So the nodular worm, it takes 40 days after infection before the worms begin to produce eggs, and can probably last about a year inside the animal. In the case of Ascaris, it takes 60 to 80 days after infection before you can begin to see eggs in the feces of your animals. And those worms can live in that pig for anywhere between 6 to 9 months. Whereas the whipworm, that one takes anywhere between 40 to 60 days after your animal initially gets infected before you start to see eggs in the feces. And the worms themselves, the adult worms, only have a lifespan of about 4 to 5 months.
Next question, can the larvae that get in the lungs cause the pig to cough?
My understanding is that yes, so some of the cough that can be seen in pigs sometimes can be due to those larvae causing little damage in the tissues of the air sacs in the lungs.
Yeah. Yushi, are you there?
Yes, I am.
A respiratory tract infection, that could do it as well. So say mold and spores and stuff, that might be able to cause a pig to cough too.
Yes, of course.
Yeah. So yeah, like Alex said, if you have a coughing pig, I wouldn't probably put it down to just parasites.
Yeah, it probably, anything like a flu kind of thing, the virus, bacteria,
Yes,
any infection
one
of
of the
the
things that
respiratory.
we teach our students at the university is that you never diagnose your patient simply on the symptoms, because many things can lead to the same symptoms. And so, yeah, the cough could be related to the worms. It could be related to other kinds of infections. Definitely the worms being there, they are causing damage to the tissue, and you have a higher risk of maybe secondary bacterial infections and other things that will take advantage of those micro lesions there. But yeah, you can't definitively say that your coughing pig is due exclusively to the worms being there.
I wanted to add just one more thing about the parasites, Alex, is that how it relates to pork production.
Right.
And so here at Rodale, our pigs have parasites. And we don't hide that information, but we do seem to be able to finish our pigs in a similar time based on industry standards. But where it becomes an issue is that you're essentially feeding parasites your grain and not the pig. The pig is or the parasites are taking that energy away, and you're reducing production. You might not even be able to tell a pig has parasites. When the infection load becomes so high, you could see they're not going to start gaining weight, but it could be low. They could be surviving and thriving, but they're stealing energy from that animal. I just want to point that out. All right, if there's no other questions, I'm going to move on then and discuss our grant that we received in 2018. So it was a USDA NEFA Organic Transition Grant-funded project titled Manure and Pasture Management to Reduce Swine Parasites in Organic Pasture Pork Production. The reason the three of us put this together is it came up at a conference one year, and just asked a group of people, hey, do we know all these kinds of information? What's the most serious thing when it comes down to pasture pork production? And it was almost unanimous that, yeah, parasites are an issue, and we need more information about how to control them. So, yeah, we developed this project to address that issue, that farmer issue, that serious issue. And you can see that's the personnel we have on it, plus 4 or 5 student technicians. It's a 3-year project. And just a brief overview of the research objectives. What is the severity of parasites on our organic pig farms? So we're going to evaluate the parasite prevalence on several farms in the United States. Second, we're going to determine the effectiveness of manure composting on eliminating swine parasites and try to understand the underlying mechanisms. So what did it? So if anybody's familiar with composting, temperature could be one of those mechanisms for destroying parasites. Third, and I'll talk more about this if nobody's familiar with the term, we're going to assess biofumigation as an approach to swine parasite control in pastures. So now we're focusing on the pasture here rather than prevalence and dealing with manure. And again, the fourth objective is going to be looking at pastures exclusively and determining the effects of grazing biofumigation pastures by organic pigs on reducing swine parasite contamination. So our first objective is, how severe is this problem in the United States? We know that for some other countries, but it's never been determined here. So our team here is going to undertake that and do a survey. And we're going to be 4 states so far included in the farm surveys. And these states are representing about 30% of national organic pig farms and over 60% of national organic pork sales. We got Pennsylvania, Wisconsin, Iowa, and Minnesota. Now, the criteria for participating farms, because when we announced that we were going to do this at conferences and grower meetings and things like that, we got a lot of interest from people that they wanted to join the survey. So we had to narrow that down for the scope of the project. We can't sample every pig farm, but they have to be organic, alternative, or at the very least provide outdoor access. Bedded floors, so no slated floors like you would find in confinement operations, and certainly no synthetic, non-organically approved dewormers. So most farmers are going to add some sort of I wouldn't say, they're going to add a natural dewormer like diatomaceous earth, and that's acceptable for the purpose of this project, or for the criteria of these participating farms. Yuchi, if you wouldn't mind describing how we're doing some of the collection.
Yes, so we went to 9 farms on these 4 states this summer, starting June 12th until August 8th. So within about 2 months. So we collect data, collect fecal samples, and bedding and also soil. Since we're not going to present bedding and soil data, so I'm going to talk about the fecal samples here. So when we collect fecal samples, farmers, they raise their pigs separately. Usually the breeding herd are housed together, and the younger pigs are housed together, older pigs are housed together. So we categorize pigs into 3 categories. The breeding herd, basically we only collect data from gestating suckers. We don't collect any data during farrowing. Actually, at the time when we collect data, no sow farrowed at that time. So that include all parodies from first parodies out all the way to parody 10 or whatever. Then we also separate the sample for younger pigs, which are about 2 months to 4 months old. Usually the pigs, they are weaned and then in the nursery for a while and then go outside or in the outdoor access farms. And then if pigs are older than 5 months old, we categorize them as finishing pigs. So anywhere from 5 months all the way to market weight. Market weight can mean 250 pounds all the way to 300 pounds. So those pigs may be at age of 6, 7, 8, even 10 months old. And then, as Alex just mentioned, we just analyzed 3 parasite species. So in the picture, you can see most of the pig we sample from, they're either on pasture or in the pen. If you look at the 2 pictures in the bottom, those are the indoor pen with outdoor access. So most of the young pigs, or finishing pigs, they are in such kind of indoor pen with outdoor access. So most of farmers, they don't put those pigs on pasture. And you can tell these pens are kind of, these farms are kind of large scale in the organic standard. They have a lot of pigs. So the very left corner, left bottom pen had about 25 pigs per pen, and the other is similar. So sometimes we even see some farmers, they put 40 pigs, finishing pigs in the pen. So if they have the breeding herd usually on pasture. You can see the middle, that's a boar on pasture, and that farm, they also put the sows in pasture. But the bird top one, you can see a farmer was feeding the animals. That farm was the only farm we saw. They put finishing pigs on pasture, and that's very kind of large scale. They put something like 60 pigs in pasture. So I know some in Pennsylvania, we have some small scale farmers, and they put their finishing pigs on pasture as well. So just give you an idea the farms we collect sample from, what they look like. Next.
Yeah, I'd say, yeah, there's a diversity of farms, like you said, that are included in the survey.
So the farm size, you know, the largest scale, they produce 500 organic pigs per year. And to the small size, they only produce something like 12, 20. So it varies.
Okay. Thank you, Yushi. And we could share some of the data that we've done. So, yeah, these are the samples that we collected from each farm. This is just giving you, there's no need to study it, but what does it say here? 185 samples collected from 9 different farms that we're analyzing for parasite prevalence and loads and
So I'll jump in here and sort of walk you through some of the preliminary results that we have. On this slide, what we have here on the x-axis, we have all the different farms grouped by the state that they were sampled from, and the colors also represent the different states. So it's all color coded. The black is Iowa, red Minnesota, blue Pennsylvania, and green Wisconsin. And this is just an indication of the fecal samples that were collected from the farm, from the individuals that were sampled. You saw the sample sizes in the previous table. This is the proportion of pigs, or the percent of pigs that were positive for any kind of a worm, whether it be Trichuris, Ascaris, or Ossiphagostomum, either a large intestinal roundworm, nodular worm, or whipworm. And we see that the numbers are pretty much all over the place. There's no real pattern by states. Some farms, 100% of the pigs were infected. Others, as low as 10% of the samples were infected. And so this was just an initial picture to see how many positives or negatives were out there. And we can see that all farms had parasites, at least one parasite. But the proportion of the herds sampled that were infected also varied quite as well. Next slide. We can break those data down, and instead of talking about the proportion of pigs that are infected, we can begin to talk about the average number of eggs per gram of feces collected that had Ossiphagostomum, the nodular worm. And again, on the x-axis, we have the different states. On the y-axis, we have the average number of eggs per gram of feces, plus the little whiskers on top of the bars are the standard error of the mean. It's not really important for now. But the different bars represent the stages of the pig. So we have either feeders, finishers, or sows. And for Ossiphagostomum, the nodular worm, we see that the farms, all 4 farms in Pennsylvania were positive for the nodular worm, and one of the 2 farms in Pennsylvania was also infected. And there really isn't a pattern in terms of differences between feeder finishers and sows at this very crude level. that we can spot. For example, the first farm in Pennsylvania, it appears that the finishers were producing more eggs per gram of feces than the sows or the feeders. Statistically, we'd probably be hard pressed to say whether there's any actual statistically significant difference between those three feeder groups. But this just sort of gives us a picture of the number of eggs that are being shed of this particular worm by the pigs in all these different farms at this particular time of the year that they were sampled. On the next slide shows similar data, but this time what we're looking at is the large intestinal roundworm, Ascaris, again presented to you by state and color-coded based on the stage of the pig, either feeder, finisher, and sows. And we can immediately see that there were a lot more farms that were positive with Ascaris. And if the pigs were infected with Ascaris, they're producing thousands of eggs per gram of feces. I mean, Ascaris has been reported that females can produce anywhere between 200,000 to 1 million eggs per day. So they're really shedding lots and lots of eggs. And if we can see again that there's a lot of variation in terms of the level of infection, if we use eggs per gram of feces as an indicator of the level of infection, we see that the numbers again are all over the place in terms of the different stages. What we don't have, unfortunately, is how these numbers vary over time, over seasons. So this is just a snapshot picture of what was happening between June and August in all these farms in all these states with this particular farm. So definitely Ascaris is a lot more abundant in terms of eggs in the feces of those pigs. And then the last slide that we have is the same, but this time is for Trichuris, the whipworms. Again, fewer farms infected with positive for these worms, and the levels of infection, at least numbers of eggs that are being shed by the pigs, is also relative to Ascaris much, much slower. And again, right now, it doesn't appear to be any obvious differences between feeders, finishers, and sows. But the question of age or stage is something that we still need to explore a little bit further.
Okay, we can ask questions, and actually, I'll ask the first question. Anybody else who wants to ask a question could type it into the chat function. But Alex, looking at the data, you said it was a snapshot in time. And given that Ascaris is so high, have you found a relationship where if Ascaris is high, and you can see on that one graph that you had that some places it was low, if Ascaris is very high, do you find that other species are going to be low or just as high?
It's difficult to interpret those eggs as an indication of the numbers of worms actually in the pigs, right? Because Ascaris is a larger worm. I mean, you're talking about, a foot-long worm, female, producing millions of eggs a day relative to a quarter-inch worm or a 2-inch worm that's going to be just by size producing a lot less eggs. So it's difficult to answer that question to see whether if one's more than the other. I think that we can maybe look at over different months and seasons to see if there are different periods of the year when one might be more common than the others. And then it's a very complicated question to answer also because there's a lot of variability between individuals. Some individuals just have natural combinations of genes that make it more resistant to a high infection level, where there are other individuals that don't have the right combinations of genes and might be more susceptible or at least resistant to parasite X, Y, or Z, any one of those three species. So it's a very difficult question to give a definitive answer to because it's a very complex picture that's going on in here.
Okay, I guess a follow-up question since there's nobody else asking, but and this could be for Yushi as well. Do we know if there's a threshold for pig infections. You know, one figure said they could be up to 4,000 eggs per gram of feces with Ascaris. If you reach 5,000, do you start really impacting growth? Do we know that yet?
I don't think anyone knows that, Rick. Um, I think, on the farm, from on-farm observation, I can tell some pigs seems very wormy, kind of doesn't grow well. It's kind of old enough, but just doesn't reach that weight. And you probably can see the reduced growth rate. And I don't know, rather than coughing, you probably see diarrhea, some pigs, but whether diarrhea is caused by parasites, diarrhea can be caused by many things. So I think reduce growth rate can be one thing, but you never know whether the pig, we see thousands eggs in that kind of sample, but we don't know 5,000 is the we don't have that. So it's depending on how many worms in and
Yes, I was just going to jump in and absolutely, it's not a definitive answer and it does vary quite a bit. One of the few things that we do understand about infectious diseases, whether it be worms or bacteria or viruses or whatever the infectious agent is that if you look at a population, a herd, that you will have few individuals that would have heavy infection loads, but the majority of your herd will either have low infection levels or not be infected at all. And this gets back to the idea of the variability between individuals in a population. They might all be the same species, but there's slightly different combinations of genes that they may have inherited from their parents and great grandparents, et cetera, that might make them more or less susceptible to infection. And, we're talking one worm, two worms, the pig is probably not going to even notice that they have anything wrong with them. But if you're getting into 1,000 worms or 10,000 worms, which sometimes you can have huge numbers, that level of infection is going to be very, very detrimental to the individual suffering from such high parasitemia. So you can imagine that, yeah, all the resources are being sucked out by these parasites, and not much is left for growing and let alone reproduction at a later stage.
Okay, there was a question that came up. Did you get to learn at any point that any farm method helped reduce certain type of worms than the other? I'm referring to those three groups of farmers that you decided to sample. Where do you see the heavy infection load? Was this predominantly observed in certain types of farms?
I probably can try to answer, and Alex and Rick, you can step in anytime. So I think in general we see a lot of worm in finishing pigs, girl finishing pigs, I should say. So compared to growing finishing pigs, looks like the sow herd is kind of relatively cleaner, and that was kind of surprised me. They look dirty, but actually in terms of parasite load, they carry very lower parasite load. So I think one probably the cells on organic farms or on the farms we sample, they are kind of old, so they may develop the immunity. And another thing I'm thinking actually, even USDA organic standard does not require the sow herd to be organic to produce organic pigs, which means farmers literally they can deworm the sow before the last third trimester. So 40 days before the sow farrow, they can deworm them with synthetic dewormers. So most farmers we visited this area don't, but they can. But I know one farm they did. They they can. So I think that two things probably result in cleaner sulfur. So the growing finishing pigs, once they are kind of 4 months, 5 months, all the way to close to market weight, we see a lot of parasites, especially Ascaris, the round worm.
Okay. There was one more question, and
If Rick, so the question was asking whether there was anything about the farmers that we sampled that might be indicative of why they have a higher or lower parasite load. So we've only just now began to plot the data, these graphs, to look at these patterns. But the farmers were given questionnaires that address questions such as the size of their production, the breed of their animals, the
good
sex
point.
of the individuals, the type of bedding that they use. And we haven't analyzed those data alongside the parasite numbers yet to answer that question.
And I think one more thing I want to add is, I feel like when the density, the stocking density is lower, you probably see lower parasite, and that's where probably.
Agreed. Actually, Yushi, thank you for reminding me of that. We do have one of the variables of the data points that we have there is the number of individuals that are kept per pen or per herd, I guess. You can tell that I'm not the farmer, I'm the parasitologist. But that that it's going to vary quite a bit in terms of what we see with the density. And yes, the more pigs per pen, if I remember correctly the data, and I will correct myself if once I look at the data later, that yeah, the more pigs per square foot, I guess, if that's what the density you want to talk about,
Right.
then there will be more. Remember, these parasites are direct oral fecal transmission. There's no intermediate host or intermediate animal that they need to go through to get back into the pig. It's oral fecal contact. Pigs are dirty. They're in contact with dirty environment all the time, and therefore they're prone to these kinds of infections. And so the more individuals that you have in a small area, the more that they're going to be defecating over each other and therefore exposing themselves to the.
And one more thing I want to add is most farmers, they use some preventative method, like say apple cider vinegar, garlic, or ginger, or Dr. Paul's product to control parasites. But we still see a lot of parasite species afloat. So I feel like those organic methods is not that effective, or the effectiveness of those organic measures to prevent parasites need to be further studied.
Okay. Given the time, we're going to keep moving on. Sheila, I see that you did ask a question, and I apologize. I'm just going to have to kindly ask that you direct your question to probably Yushi for that afterwards, and I'll have her contact info provided at the end. But we only have about 7 minutes left, so I want to make sure we cover it. So our objective 2, here's Rodale's large-scale composting operation. This is the operation that I run here at Rodale, and it's high temperatures we generate in these this type of composting operation. So during this objective, we're going to take the swine bed packed manure, compost it to completion, and we're going to collect samples before we compost it, and then weekly for 4 weeks during composting. And we're going to try to measure, egg counts in those samples. So here's a graph of time and temperature, and we have 2 trials that we've run already. One on the left was done early in the year in January, and then the one on the right was done in middle of summer. And so you can see they're very different. The temperature scales are the same, and the ambient air temperature is going to be the light gray. Those red dots are when I'm turning with that machine that you saw in the background. And so let's just look at the one, pile 80, the one on the right. You have temperatures above 60 degrees Fahrenheit for a sustained period of time. And in the composting industry, that kind of thing is acceptable for destroying all human pathogens, all plant pathogens, and plant nematodes. And so we predicted that that was going to be what destroyed. That high temperature is going to be what's destroying the swine parasites, but nobody's ever looked at it or documented it. So here's a graph of time and the the eggs per gram that was adjusted there just to put everything on the same figure. So because you saw the um, Ascaris is going to be way higher, but it's just adjusting it so we could see it. But so things I would have expected some of these figures here, these lines, to have a dramatic decline within 1 week, 2 week, 4 week for sure. But it didn't really show us that. So we repeated it again, and during that time, temperatures weren't as high as I would have preferred from a composter perspective. But we repeat that again, and you do see that dramatic decline in the eggs that we're detecting in the compost samples. And so by week 4, what is it, the Trichuris, that didn't seem to respond at all, which was a little odd, but Ascaris and Ossifagostomum, within 1 week, Ossifagostomum was no longer detectable. And Ascaris dramatically dropped within 2 weeks, and it continued to drop. So that's promising. We're going to repeat this, and you'll be able to see that data in the coming year. All right, so what we're going to be doing in the lab in the meantime, that we're just now starting this, is we're going to conduct bioassays to determine if certain time and temperature conditions are going to be enough to inactivate or destroy these parasite eggs. And we'll be able to assess viability. Do they germinate or they don't? Because when we're looking at them under a microscope, it's not easy to tell if an egg is viable or not. But if we allow it to germinate, that might tell a different story. I'm going to skip questions just for a second and answer them at the end, given the time, and go on to objectives combined 3 and 4, the biofumigation. And I'm going to simply say that certain plants, particularly mustard plants in agriculture, that when they break down, they produce a gas. And the gas is similar to mustard gas, isothiocyanate, cyanide essentially, but it's a gas. And what we've been able to do in agriculture, not as effective as other means, but it's proven, or not proven, but there's a lot of documentation to say it can work. So you have your pasture there of mustard, and you chop it, you incorporate it as quickly as you can, you pack it down, and you're trapping the gas. And in this case, Cornell was using it to treat either nematodes or plant pathogens. And so we're looking at a few different species: yellow mustard, rapeseed, Alice white clover, and ladino clover in this. And that type of work is still ongoing. But that's going to bring us into, this is what it looks like. This is what we've done. So this is the kind of the field work. So we have our stand. Here's Alex and his team. We're doing artificial inoculation with fresh pig feces. And then we wait a week and we mow it down with a flail mower, which drops the material right in place. And then till it in, and all this is happening one step after another as fast as we possibly can. So within an hour is what we're trying for. And then we pack it in with this Cultipacker. And then afterwards, we're going to do weekly soil sampling for 4 weeks and try to see if there's going to be a decline in egg numbers. And again, I'm just going to quickly brief over the research objective number 4 is taking the biofumigation more to a practical standpoint and testing this out in the pig pastures with live animals. So the difference here now is that we're going to be examining is mechanical incorporation of plant residues versus biological. So we'll have it divided into, and you can see the 2 different pictures here where this is me rototilling it, and then your end product is there, me packing it in. But can we have the pigs do that? And this is the pigs going after turnips. Can we have the pigs do that and then pack it in and destroy it? And so we'll aggressively move them through the pastures. And so this is what the field plot looks like. It's a little convoluted, but everything's going to be divided up here at Rodale Institute in our pastures. And so they're going to be moved through paddocks, 4 different paddocks. And then we're going, after they get moved through, we'll be chopping, one side of it will get the mechanical incorporation of chopping, mowing, and tilling it in, and then the other side is just what the pigs did. All right, so with the last minute or so, I'd like to just say thank you, and if there's any other questions, we could address those now. I appreciate everybody attending this webinar. Sorry I had to go through that pretty fast, but actually, Sheila, since your question's up there, usually if you want to address, is anyone selectively breeding these individuals less prone to parasites, you would probably have a better idea than I would.
I don't think anyone has done that. And as I said, the cells are kind of, immune, more immune. And I'm thinking as animals getting older, they probably have more resistance to parasites. But regardless, the younger animals, they get it. So I don't know, if you look at different species, different animals, if we do that, we may find something, we may make some progress. But so far, I don't think anyone has done that.
Do you see any potential at all? And Alex, you from
Yeah,
immunity or resistance perspective, is there a potential?
I believe so. I believe so, but just there is. I believe there
There's
is. I
potential
believe
for that.
different species, different, even within the species, different animals. If we select for that, we may see some progress. But parasite is very complex. It's
Yeah.
not like selecting for growth performance. It's depending on the host, the parasite. It's a lot of things there.
Okay. Alex, you were going to add something?
Yeah, no, I was just gonna say I agree with you. I think the potential is there. I think it's just gonna take a lot of thinking about what specifically, what characters can you possibly identify in an individual that is more immune, has a healthier immune system than the other. I think that's good. That's the challenge, right? You can just look at the individual and maybe if
There,
they're sickly and thin, you can tell not to breed those individuals. And I think that farmers probably do that already, to not breed the sickly looking individuals, the more healthy looking individuals. But yeah, no, that the genetics is a powerful thing that is underexplored.
right, in Denmark, they did such a study years ago. They inoculated the pigs with parasites, and then they measure the antibody produced. Some animals stimulate stronger immunity, produce a lot of antibody, but some others did not. And then you probably think, if I select that, that may help, but that may not necessarily mean to eliminate. That may, help, but how efficient that will be. So there's a potential there, but
And then the picture also gets complicated when you start to look at differences between the genders. So, for example, the boars, intact males are probably going to be have a weaker immune response as a result of them having higher levels of testosterone, which are immunosuppressive in nature. So, in nature, in just about every species of animal that I've studied in my career, intact males tend to have higher worm burdens on account of their higher levels of testosterone, which are immunosuppressive, which means that their immune system can't fight those diseases as well, relative to other genders. And then when you talk about females, sows, for example, for the nodular worm, there are reports of there being this rise around the time of parturition or time of birth, because again, the immune system becomes compromised around this very important period in that sow's life stage, that she becomes immunocompromised, and then you see a spike in the levels of infection in periparturient females or females that have just recently given birth. This is commonly seen in sheep and goats, but it's also been reported in the species that infected pigs as well. So the picture is very, very complicated because of all of these different variables.
right, and plus the heritability, what's the heritability of the immune system
Exactly, right.
is another.
Yeah, we don't know. Right.
Okay. Well, thank you to my panelists, Alex and Yushi. Unless anybody has a question in the next few seconds, I think we'll call it a day. And again, to my attendees, thank you very much for attending. I hope you gained something from this. Take care, everyone.
Bye-bye.
Thank you. Bye.